Assalamu alaikum dear student, I'm Dr. Uzma and today we are going to discuss enzymes. There are two fundamental conditions of life. First, self-replicate, and the second is must be able to catalyze the chemical reaction efficiently and selectively. For example, when we consume the normal table sugar, that is sucrose, in the form of food and sweetener and drink, the conversion of the sucrose to CO2 and H2O, that is the carbon dioxide and water, in the presence of oxygen is highly exergonic process, releasing the free energy that we can use to think, move, taste and see. However, a bag of sugar can remain in the shelf for years without any conversion to CO2 and H2O. This thermodynamic process is also favorable, but it is very slow. Without catalysis, chemical reactions such as the sucrose oxidation could not occur in useful time scale and thus could not sustain life. History. In the late 1700s, biological catalysis was first recognized and described in studies on the digestion of meat by secretion of the stomach, and the research continued in the 1800s with examination of this conversion of starch to sugar by saliva and various plant extracts. In the 1850s, Louis Pasteur concluded that fermentation of sugar into alcohol by yeast is catalyzed by ferments. He postulated that these ferments were inseparable from the structure of living yeast cell. This view called vitalism. In 1879, Edward Buscher discovered the yeast extract could ferment sugar to alcohol, providing that fermentation was molecules that continued to function when removed from the cell. Frederick W. Koenig called these molecules enzymes. 1926, the isolation and crystallization of urease by James Sumner provide a breakthrough in early enzyme study. Sumner found that the urease crystal coincidentally of protein, and he postulated that all enzymes are protein in the absence of other example. In the absence of other, like it is a very controversial part. Okay? In 1930s was Sumner conclusion widely accepted after John Northrop and Moses Kunitz crystallized pepsin, trypsin and other digestive enzymes and found the also be a protein. During this period, J.B.S. Haldane wrote a treatise entitled Haldane made the remarkable suggestion that weak bonding interaction between an enzyme and its substrate might be used to catalyze a reaction. This insight lies at the heart of our current understanding of enzymatic catalysis. Since the later part of the 20th century, research on enzyme has been intensive. It has led to the purification of thousands of enzymes, elucidation of the structure and the chemical mechanism of many of them, and a general understanding of how enzymes work. So let's see what are enzymes. Enzymes are biological catalysts. Most enzymes are three-dimensional globular protein like the tertiary and the quaternary structure. Okay? They increase the rate of chemical reaction taking place in a living cell. It means the enzymes, which are the biocatalysts, they speed up the chemical reaction without itself being consumed. Such reactions are called enzyme catalyzed reaction. Okay? The features of enzyme. They are the most remarkable and highly specialized protein. They have extraordinary catalytic power greater than synthetic and organic catalysts. They have high specificity for the substrate. They accelerate the chemical reaction tremendously. They function in aqueous solution under very mild condition of temperature and pressure. Enzymes are central of every biochemical process. They act as organized sequences, hundreds of the stepwise reaction that degrade nutrients molecule and conserve and transform into chemical energy. They help in making the biological macromolecules from simple precursor. Enzyme deficiency and overactivity lead to the diseases. Therefore, measurement of activity of enzyme in plasma, erythrocyte or tissue sample are important in the diagnosis of certain illness. Many drugs act through interaction with the enzymes. Enzymes are tools in chemical engineering, food technology, and agriculture. Note: all enzymes are protein except ribozymes. On the basis of enzymes can be divided into two types: the simple enzyme and the complex enzyme. Simple enzyme, which is made up of only one protein, example urease. Complex enzyme, it contains the protein as well as the non-protein part. The protein part of the complex enzyme is known as the apoenzyme or apoprotein. The non-protein part is known as a cofactor. The cofactor may be of organic type or a metal ion. The organic molecule can also be known as the metallo-organic molecule coenzyme, and it contains a metal ion which is act as an activator. You can see there is an inactive protein and there is a cofactor. But when the cofactor is bound to the active site, the cofactor binding activates the protein, and without the cofactor attached, the protein is not active. Some enzymes bind cofactor more tightly than others. Similarly, when cofactor bind is bound to the enzyme more tightly, that is difficult to remove without damaging of the enzyme. It is known as the prosthetic group. Inorganic cofactors. These are the inorganic molecules required for the proper activity of the enzymes. For example, enzymes like carbonic anhydrase, which require Zn2 plus for its activity. Hexokinase has a cofactor which required magnesium 2 plus for its activity. Organic cofactor. These are the organic molecules required for the proper functioning of the enzyme. For example, the glycogen phosphorylase requires a small organic molecule pyridoxal phosphate. Diagram, you see the type of organic cofactors like the prosthetic group and the coenzyme. Okay, what is the prosthetic group? A prosthetic group is a tightly bound organic cofactor. Example, the flavin, heme group and biotin. What is the coenzyme? A coenzyme is loosely bound organic cofactor like NAD+. Let's see what is the inorganic elements that serve as a cofactor for enzyme. There is a list of these enzymes. Please kindly learn. Cu2+, which is the cytochrome oxidase, Fe2 plus or Fe3 plus, which is the cytochrome oxidase, catalase, perioxidase, K plus, pyruvate kinase. These are the cofactor which requires the enzyme. Okay, inorganic element that serve as a cofactor for enzymes. Magnesium 2 plus, hexokinase, glucose 6 phosphatase and pyruvate kinase. Mn2 plus, arginase, ribonucleotide reductase. MO, dinitrogenase. Ni2 plus, urease. Se, glutathione peroxidase. Zn2 plus, carbonic anhydrase, alcohol, dehydrogenase, carboxypeptidase A and B. Some coenzymes that serve as a transient carrier for a specific atom or functional group. These are the coenzymes. These are the example of chemical group that they transferred and the dietary precursor in mammal. Let's take a look. Biocytin It requires example of CO2 and its dietary precursor is biotin, coenzyme A, and its chemical group is acyl group, and its dietary precursor is pantothenic acid and other compound. The coenzyme is 5' deoxyadenosyl cobalamin, which is known as the coenzyme B12, and its chemical group is hydrogen atom and alkyl group, and its dietary precursor is vitamin B12. Like the coenzyme flavin adenine dinucleotide, its chemical group is the electrons, and its dietary precursor is riboflavin, which is the vitamin B2. Lipoic, its chemical group is electron and acyl group, and they are not required in diet. Nicotinamide adenine dinucleotide, its chemical group is hydride ion, and it is required for dietary precursor of nicotinic acid, which is also known as the niacin. Pyridoxal phosphate, it requires chemical group amino acid, and it is required for vitamin B6, which is also known as the pyridoxine. Tetrahydrofolate, and it requires one carbon group, and it is required for dietary precursor of folate. Thymine pyrophosphate, and its chemical group is aldehydes, and its dietary precursor is thymine, which is known as the vitamin B1. Thank you so much.